Hello everyone, my name is Nuttachat Wisittipanit. I am from Luang University in the Department of Material Engineering. So, today I'm going to present the topics of Lexomo, rapid typing and characterization tool for whole genome sequencing data of Salmonella. So this is a presentation for the Bioinformatics 2023. So before we go into the details, right? Outbreak events, involving the foodborne pathogens such as Salmonella enterica happen more frequently nowadays. When such event occurs, in our life, either with the endemics or pandemic level, they must be dealt with swiftly and quickly, in order to prevent future spreads and get people sick. Usually, it's a gastroenteritis. People will get diarrhea and may lead to death. So this research presents the bioinformatic tools, based on the whole genome sequencing data called the Lexomo. This Lexomo it can analyze a group of genome assembly of Salmonella enterica, in a short period of time, and also it can provide information for healthcare professionals. Is why when they want to make a decisions in order to deal with these outbreak events in timely manner. So I mean, they can use this tool to track to the origin of the Salmonella species or serotype, and then pinpoint exact serotype and then inform the related individuals, in order to make actions. So Lexomo is a Linux-based bioinformatic tool. It's a fast decision-aiding tool using the whole genome sequencing or WGS data of Salmonella enterica, for the investigation of an outbreak. So the data that we use in this Lexomo tool, is a genome assemblies. It is in the FASTA format, FASTA, and input to the program is a folder. It's a folder containing a group of genome assembly files. And the requirement is that all genome should be belonging to the Salmonella enterica species. All right. But if it's not in the Salmonella enterica species, the tool can also detect that as well. But Lexomo is a command line program, that can handle multiple WGS analyzed in one go. It can analyze maybe 100 or maybe 200 genomes. And then it's quite quick. So the analysis process include the genome-based typing, profiling of antimicrobial resistance, phylogenetic tree constructions, and CRISPR analysis. Also, it can find CRISPR1 and CRISPR2 locus. The typing process comprises of traditional Salmonella serotyping, multi-locus sequence typing, or MLST. And then also CG MLST, which is a core genome multi-locus sequence typing. Also, the tool can also find the AMR profilings. So it means AMR mean antimicrobial resistance. This tool can determine the antibiotics classes found associated with genes in the genome of Salmonella enterica. So this is a crucial information in order for professional to treat the patients with correct antibiotic drugs. It will tell you the antibiotic class and then also the antibiotic genes of the genome that one particular serotype has. Okay, so the AMR process tries to determine the antibody class found associated with genes, okay, within the genome of Salmonella enterica. Okay, so and then the tool also can also build the phylogenetic tree, all right, that can help track the exact source of the outbreak events using the alignments of core genome with the reference strain of LT2 Salmonella. This is useful, okay, because it can pinpoint the exact strains that can cause the outbreak event. Okay, so the professional can quickly go into that source and then eliminate maybe the animals. Okay, or maybe the plants, okay, such as onions, maybe some tomatoes, okay, that is the source of the outbreaks. Okay, also the CRISPR analysis also include the number of spacers in both CRISPR1 and CRISPR2 locus, okay, and also their lengths. Okay, so the output of all the procedure consists of one summary file in LSX or Excel, okay, and also the CSV format. Pie chart of serotyping, MLST and CG-MLST, okay, an image of phylogenetic tree, okay, and also CRISPR analysis results as well. All right. So the input that we use to demonstrate these two capabilities, okay, is we have a total of 12 genome of Salmonella enterica, okay. So this table show the cellular, okay, confirmed cellular, the test in the lab, okay, and also the collection years, also the location of the source of the Salmonella genome. Okay, we have from Thailand, from Switzerland, from United States, from United Kingdom, okay, and also from Germany. Okay. Okay, so the output of the two, this is the part of the output. Okay, the first part. Okay, it shows the assembly name, right? Assembly name, and also the serotype. Okay, get from the two serotype. This one we get I4512, right? And then the MLST, okay, or multilocus sequence typing, and CG MLST, okay, or core genome multilocus sequence typings, okay, and also the cellular, okay, cellular CG MLST, okay, that which is try to determine the serotype from the core genome, okay, and then also the cellular group, also the AMR class, okay, AMR class tell you the class of antibiotics drugs, okay, that the genome is resistant to, okay. All right, and then also we have okay, also this is a CRISPR analysis results, okay. We have 12, right? We have 12 genome, right? And then for each genome we show the number of spacer for CRISPR1 and also number of spacer in CRISPR2 locus, okay, and also the length of CRISPR1 locus and the length of CRISPR2 locus as well, okay. CRISPR is like maybe tool for modern gene editing, okay. So if you want to do a gene editing nowadays, okay, people professional in bioinformatics, okay, try to first use the CRISPR tool, okay, to the edit genome anywhere we want, okay. This is a quite powerful tool. But now we will show only this result, okay. So this is the pie chart of all the serotype, okay, that the tool detects, okay. It has relative radiance, for example, okay, Enteridis, okay, Kentucky, Typhimurium, okay, or Clearfield, right, each 8%. So this is show showing the cytogenetic tree, okay, using the Salmonella LT2 as a reference genome. Okay, this one, right? This one, and then other is the show in besides this reference genome. All right. So these two, okay, this three, as I said before, okay, can be used to track the origin of the outbreak strain. All right. And then what? Okay, so the conclusions. Lexamo is a bioinformatics tool, okay, and also decision aiding tool in the rapid Salmonella enterica investigation to mainly aid healthcare professionals and experts. Okay, in the case of an outbreak event, okay, it takes the genome assembly of Salmonella enterica and produce outputs that report serotype, ST, CGMLST, Silova, Silogroup, MR classes, and also CRISPR analysis results and pie chart of serotypes, as well as the cytogenetic tree, okay, that can track the exact source of the exact outbreak source. Okay, future update. This tool is not yet complete. Okay, future updates maybe include the determining of Salmonella pathogenicity islands. Okay, and more in-depth of CRISPR analysis results as well. Okay, maybe I want to include the sequence of spacer, the name of the spacer that in the locus of each CRISPR1 and CRISPR2, like that. Okay, it may be take maybe a few more months. Okay, before this tool is complete, and then I will be publish it. Okay, so thank you very much for your attentions.